This reaction yielded hydrogen peroxide, H2O2, and aqueous hemoglobin, Met-HB*H2O, and the oxidation product of aquapentacyanophorate 2, i.e., 2-. The reaction was found to undergo overall second-order kinetics with a first order in each oxidant, PO2, and reductant, 3-, respectively. The results declared that the structures of the reactants such as protein and external donor control the kinetics of the electron transfer with an inner sphere mechanism that involves direct electron transfer from the aquapentacyanophorate 2, to bound dioxygen that yields peroxide, subsequently. Another study surfaced the effect of binding sites and protonation on the kinetics of the electron transfer reactions of blue copper proteins. The oxidants with different binding sites such as 3-, 3-, and 3+ were used to oxidize parsley plastocyanin. In each reaction, regardless of the binding sites, and prior to electron transfer, a strong association between protein and complex occurs. The variation in the binding sites varied the reduction potential and affected the rate of electron transfer, consequently. The reductant, plastocyanin, is a copper protein that consisted of type 1 copper, which is involved in electron transport from photosystem II to photosystem I at the surface of the thylakoid membrane. A single copper here utilizes oxidation states 1 and 2. The structure of poplar plastocyanin peaky contains Chu2, coordinated with 2 histidines, 1 cysteine, and 1 methionine in a distorted tetrahedral arrangement. It has always been of interest to probe the details of the transfer of electrons and protons because of successfully unveiling strategies of energy conversion in both of the fields, biology and chemistry. The energies as well as mechanism are strongly influenced by the coupling of electron and proton transfer. This defines the need to build up multiple redox equivalents to carry out those reactions that involve multi-electrons. This also explains those mechanistic pathways through which electron and proton transfer occur simultaneously to avoid intermediates of high energy. The theoretical background of the proton-coupled electron transfer reactions in solutions and proteins and electrochemistry was reviewed and discussed. The theoretical treatment was based on the calculations of multi-state continuum theory wherein the solvent provides dielectric continuum, the solute is treated as a multi-state valence bond model, and quantum mechanical approach is used for transferred proton or hydrogen nucleus. The rate expression of electronically non-adiabatic electron transfer and proton-coupled electron transfer depends upon the reorganization energies of solute, inner sphere, and solvent, outer sphere, and also upon electronic coupling. For proton-coupled electron transfer, this is the average of the proton vibrational wave functions of the reactants and the products. The compensation of the smaller outer sphere solvent reorganization energy for proton-coupled electron transfer by the larger energy needed to coupling for electron transfer appears with a similar rate for both electron transfer and proton-coupled electron transfer in calculations. A comparative theoretical study supported the reviewed outcomes through the proton-coupled electron transfer, single proton transfer, and single electron transfer reactions in iron biimidazoline complexes. The oxidation of organic compounds by a number of oxidants either of inorganic nature or organic nature has been of interest. These redox reactions are usually catalyzed by transition metals. The kinetics of the oxidation of pyridinycarbaldehyde isonocoshenol hydrazone to isonocoshenol picolinoyl hydrazine was studied, and the mechanism was proposed in the view of results obtained in aqueous solution. The reaction was catalyzed by iron 3. Advanced oxidation processes, OPS, are used to remove pollutants/contaminants such as organic and inorganic compounds from water and wastewater by oxidation of these unwanted compounds. The process involves a number of chemical reactions consisting of oxidants such as ozone, O3, hydrogen peroxide, H2O2, and UV light with the addition of catalysts that may lead to yield hydroxyl radical, O, which degrades such pollutants, dyes and organic compounds, for example.